Welcome everyone. So today we are going to study the topic skeletal muscles. Let's quickly talk about the structure of a skeletal muscle. So a bone is connected via a tendon to a muscles. Okay, this is tendon, this is a muscle, skeletal muscle, this is the bone. So this muscle circular, like when we cut through the muscle, it is circular. The outer is known as the lamination, or the wall of the muscle is known as epimysium. Muscle is further made up of many repeating units. These are also like long filaments running inside the muscle. These are known as fascicles. Okay. This is further a magnified fascicle. So this fascicle, the covering of a fascicle is known as perimysium. So this fascicle is further made up of like many small repeating, again long filament like structures running inside a fascicle known as muscle fiber. Okay, this is a magnified muscle fiber. Again, this muscle fiber will have the blood vessels on it for the conduction of the nutrients and the energy that is required for the muscle contraction. This muscle fiber is further made up of many small repeating units known as myofibrils. The covering of this muscle fiber is known as sarcolemma. And the cytoplasm kind of thing inside the sarcolemma is known as sarcoplasm. So basically, myofibrils are the smallest repeating units of a muscle that constitutes a muscle. Muscle further made up of fascicle, fascicle further made up of muscle fiber, and muscle fiber further made up of myofibrils. Now let's see what does a muscle fiber, a myofibril magnified, looks like. So okay, this is a muscle fiber. Muscle fiber is made up of many fascicles, and when we cut through a myofibril, these are the myofibrils. When we cut through a magnified piece of myofibril, we see striations like this. Like there are various bands, like dark bands, and they're having translucent kind of line in between. Then we have light bands. These are the dark bands. These are the light bands. Again, we have the dark bands, then a line, then dark bands, then light bands. Okay, so this kind of striated appearance we get through a myofibril. So myofibril, because of this striated appearance of myofibril, skeletal muscles are also known as striated muscles because of its appearance. Now let's study why do we get such kind of striations inside a myofibril. A myofibril is made up of two kind of filaments. These are the actin filaments and myosin filaments. Actin and myosin are two type of proteins that make the myofibrils. This is the myofibril. Okay, so a myofibril have two kind of filaments, actin filament and myosin filament. These are proteins. So actin filaments are the light filaments, thin filaments, and these myosin filaments, these are the thick filaments. These are attached in center by the Z line. This is a sarcomere. A sarcomere is the repeating unit of a myofibril. a myofibril. This is a myofibril. It is made up of many sarcomere. Okay, so now let's see what does a magnified sarcomere would look like. So okay, this is a sarcomere. Let's supposing I say this is a sarcomere. Sarcomere is defined by its walls by adjacent Z lines. These are the lines. Two adjacent Z lines constitutes a single sarcomere. So this is the whole myofibril. We have many Z lines. These Z lines are not basically straight. These are the zigzag kind of lines. That's why named as the Z lines. Okay, so these Z lines are to be considered as the center for a thin filament. Like, let's suppose this is one single sarcomere, sarcomere number 1, and this is sarcomere number 2. So the thin filaments, these are the thin filaments. These thin filaments have their centers at this Z line. Okay, and this band that is made up of only the thin filaments. These thin filaments are actin filaments made up of actin protein, and thick filaments are made up of myosin. So this actin filament wala band जो है, this is known as I band. एक sarcomere के अंदर we have various kind of bands. These are the thick filaments. These are the thin filaments. Okay, so we have different kind of bands. These are the I band, and this is the, let's write it down here, this is a A band. So okay, now let's say that a sarcomere is made up of two half I band. Like, look at this. This is half of the I band and this is half of the I band. So a sarcomere is made up of two half I band and one whole A band. Okay, so now you guys can relate this. These are the Z lines. Half of the I band is in one sarcomere. Other half of the I band is in another sarcomere. So we are talking about a single sarcomere is made up of two half I bands and a single A band. So okay, this is a sarcomere divided by Z lines having the A and the I band. I band consists of the thin filaments. A band purely consists of the thick filaments and somewhat little portions of the I band or the thin filaments. So these thin filaments are made up of actin and these thick filaments are made up of myosin. Now, how you guys can remember that what are these thick filaments made up of and how to remember that thin filaments are made up of actin? The trick is that thick filament always lays at the center. Like, these are not attached to the sarcolemma. You remember sarcolemma is the wall of the myofibril. The thick filaments are not attached to the like Z lines also. These lay down in the center, so these have the heads on it, head kind of projections. These are the myosin heads. So myosin heads are at the center. These are the thick filaments. Therefore, thick filaments are made up of myosin, having myosin heads. Okay, and the actin filament or the thin filament, these are attached to the Z lines. Now, there is also a M line at the center of the thick filament that will help holding all these thick filaments together inside the muscle. This line is known as M line. So okay, actin filament and myosin filament. I have already told that myosin filament have the myosin heads all over the filament, and this actin, it is further made up of three kind of proteins. Let's draw the structure. It's also in the form of helical structure, two road structures of circular actin protein. These circular proteins are the actin protein, having a line of tropomyosin. So this tropomyosin, let's suppose this is the actin filament, the tropomyosin laying above the tropomyosin laying above the actin filament will block the attachment site for the myosin heads. These are the myosin heads. This is tropomyosin. And this is the troponin. So, okay, so this tropomyosin will block the site for the attachment of the myosin head, that is the troponin laying on the actin protein. Okay, I'm sorry, these are the actin myosin binding site. So this tropomyosin blocks the actin myosin binding site, and there lays these two small dot kind of things. This is known as troponin. This is troponin. This is tropomyosin. This whole thing constitutes of the actin filament or the thin filament. Okay, so this thin filament is the filament. You remember, this is the Z line inside an I band. We have these thin filaments. So this thin filament or the actin filament looks like somewhat like this: circular actin filaments, proteins, fibrous tropomyosin, and circular troponin. So this tropomyosin blocks the actin and myosin binding sites by the help of troponin and tropomyosin. So now let's study that how does these myosin heads binds with these actin and myosin binding site, and how does this blocking tropomyosin opens the gate for the binding of myosin head and actin. So this is how does the arrangement of thick and thin filament inside a myofibril or inside a sarcomere would look like. These are the Z lines, these are the thick filaments, these are the thin filaments, these are the Z lines, and thin filaments made up of actin, troponin, and tropomyosin. So let's say these are the troponin. So this tropomyosin blocks the activated site for the binding of the myosin head with the actin filament, with the actin protein. These are the sites. Now the covering of a myofibril the layer, the wall of the myofibril is known as sarcolemma. Remember? Whenever our muscle gets a stimulus, like our neurons get stimulated from our nerve endings, we get the certain chemicals that activates the sarcolemma to secrete calcium ions. Calcium, 2 positive ions. So with the help of these calcium 2 positive ions, the tropomyosin that is like blocking the activated site for the binding of myosin head with the actin protein. It displaces and it allows the actin myosin binding site to further carry forward the muscle contraction. Like whenever the sarcolemma secretes Ca2 positive ions, these tropomyosin gets activated and it just displaces from its original resting position, allowing the binding of myosin heads with the actin protein. Like these are the actin proteins, and this is, let's suppose this is the tropomyosin. So whenever Ca2 positive ions will be there around it, the tropomyosin displaces, allowing the binding site, actin and myosin binding site is now available for binding of the myosin heads. And what happens is that ATP molecule gets converted into ADP and inorganic phosphate, and energy is released. So this energy helps the bending of this myosin head. Now this is somewhat this movement somewhat looks like whenever ATP reaches the myosin head starts binding with the actin myosin binding site present on the actin protein. So ATP released, energy was consumed, ATP is now converted into ADP and inorganic phosphate, and bending occurs, bending of this myosin head. Now what happens is, so this is the Z line, these are the thin filaments, and this is the thick filament. So what I'm saying is that these myosin heads will move. This is the thick filament originally. After contraction, wait for a second, let's see, this is the thick filament, these are the thin filaments. This is the original position. Now after contraction, what will happen is, this is the thick filament and these are the Z lines. So during a contraction, Z lines come together, come close, and the thin filaments move close to each other, moves over this myosin thick filament, and the Z lines come closer. So let's say that this is our sarcomere, these are the Z lines, these are the thick filaments, these are the thin filaments. A contracted sarcomere would increase in its width and its diameter. Its diameter is obviously going to increase with the contraction. The distance between two adjacent Z lines, here it was this much, now it's just this much, would increase. Now also to keep in mind, we had two kind of bands, I band and the A band. A band was this thing, like this purely thick filament, the area of the thick filament only, like not this one. This one is considered inside the, this is the A band. I band will have only the thin filaments. I band will have only thin filaments. A band will have thin, somewhat of thin, like these thin filaments, purely thick filaments. So during the contraction, I band, you will notice that I band has shortened, whereas the length of the A band has remained the same. Okay, during contraction, the length of the A band remains same as it was initially, and I band has contracted. The thin filaments have come closer to each other. The distance between the Z line has decreased. These are the Z lines, has decreased. So the length of a sarcomere has decreased. And in this way, the stimulation was due to Ca2 positive ions. Okay, so Ca2 positive ions was secreted by our sarcolemma, the wall of the whole